Good morning, everyone. My name is Erin Tranfield. I'm the head of electron microscopy at the Instituto Gulbenkian de Ciencia just outside Lisbon. So this morning in the multimodal imaging session, you're going to be stolen a little bit into the world of electron microscopy and how we combine this with light microscopy. In this vein, it is really my great pleasure to introduce our very first speaker this morning, Dr. Paul Verkade. Now, I asked our speakers to give us a little bit of information about themselves and a fun fact, because it is Friday morning and it has been a long week. So I hope you will enjoy these little introductions because each one of them contains a little nugget of mischief, shall we say. So to introduce Paul, Paul did his PhD in Utrecht, the Netherlands, following a postdoc at EMBL in Heidelberg, and starting a new EM facility at the Max Planck Institute in Dresden. Paul ultimately moved to Bristol, UK in 2006. Paul has remained in Bristol as a professor of bioimaging in the School of Biochemistry at the University of Bristol. Now, Paul says he always does everything in 5-year stints. First, he did his teaching degree, then his university, then his PhD. Through EMBL, even the tech development as a group leader in Dresden. But according to Paul, he has failed miserably since moving to the UK because he's been in Bristol for much more than 5 years. So it is with pleasure that I think Paul, well, at least for me, that Paul will introduce some of this amazing work he's done during this time of failure and some of the multimodal imaging that he is doing in Bristol at this time. Paul, the floor is yours. Take it away, and thank you for joining us here at SPALM.
Excellent. Thank you very much, Erin, for the invitation and also for the kind words. Yeah, I have never managed to, so to say, leave the UK after I moved here, but who knows what's going to happen in the next month or so when Brexit finally gets decided. We'll see. I'm going to share my screen. Yep. There we go. Okay, so what I'm going to do is talk about correlative light electron microscopy strategies, which we use in the lab for our life science research. And what I'll do is, first of all, I'll talk a little bit about CLEM, or correlative light electron microscopy, just what we mean by that. Why would you do something like that? And then I'll talk about that there are many, many workflows you can use for CLEM. And today I will only highlight two, maybe just briefly mention a third one of what we do in the lab. So what do we call correlative light electron microscopy? Well, it's really the observation and the analysis of one and the same event by both a light and electron microscopy technique. So that's not for all technologies that's possible, but what we try to aim in our research is really look at something in the light microscope and trace back and analyze exactly that same event in the electron microscope. So why would we do that? Well, both these technologies, light microscopy, electron microscopy, they have their own advantages and disadvantages, limitations. So in light microscopy, if you look down the microscope, you have many cells you can look at. So you can pick and choose which ones look the best, which best resemble what you want to image. That means that you can also do live imaging. So you have a history of events in the microscope. Here, for instance, most cells are labeled with a red dye and a green dye, but there's one cell here, for instance, that is an outlier. It also expresses a blue fluorescent protein in this case, and that means that in the light microscope, you can also identify unique cells. And if I have the time, I will also come back to where light microscopy is really an aid, not as much as in live imaging, which we use for Cryo CLEM. Now, if we then look at the electron microscopy, we get the higher resolution of the electron microscope. which is nice, but even more important, we have the reference space. So in light microscopy, we only see the proteins that we label with our fluorophores, but the structural surrounding we cannot see. And that's what we get really in the electron microscope. But of course, electron microscopy is looking at that material. We can't do any live imaging in electron microscopy. So if we combine those two, we may get much more out of the technologies themselves. Now, there's many ways you can do correlative light electron microscopy. You can write down all kinds of technologies and see how you can combine them. You can do that with pictures as well. There's, however, one important thing that you should take home from this is it's all nice to have these technologies, but the very first thing you need to do is look at your own biological question. What is the biological question that I want to answer? And that drives what kind of technology you will use. Correlative microscopy is really very hot at the moment, and everybody wants to do it. But we sometimes get people that ask us, how many mitochondria are there in a cell? That is something you don't need to solve with correlative microscopy. That's something you can easily do just with light microscopy if you have a very good dye and super resolution, or with electron microscopy. You don't need that combination. So it's sometimes really where you have to think, do I need correlative microscopy to answer this biological question? And so what I'll do today is I'll highlight two projects that are very different and have required different solutions and different approaches for correlative microscopy to answer that biological question. The first one is about the endocytosis of material from the outside of the cell. So we have the EGF receptor here in red, and that is internalized by the same method, clathrin-coated pits and endocytosis, as the transferrin receptor in green here. But at a certain point, they are internalized together, and then in the early endosome, they are segregated. The EGF receptor is degraded, the transferrin receptor recycles back to the plasma membrane. And that's something we would like to understand better, what's going on in that segregation event. So of course, what you start out with is doing live light microscopy. What's exactly happening? And so we have added here, so this is one cell, and I realize the resolution is not great here. We have one cell that has taken up transferrin Alexa 488, so the green dye, and EGF coupled to Alexa 594, the red dye. And we see those structures moving. And I'll show you some snapshots from that. There are two here. There's one here, which is at 7 or 8 seconds. And about half a minute later, we have another one. And you can see this long extension, which is mainly green. So there's a green-red structure, a red part, and then there's this long green tube. And this green tube, that's the transferrin, which we think is recycling. Now, that's the part we would like to capture. How do you capture something like that? We have to fix that moment in time. And so to fix that moment in time, we basically have two ways. We can put on chemicals like formaldehyde, glutaraldehyde to stop the process, or we can do cryofixation. Now, chemical fixation is much easier. Cryofixation, you need special technology. So we started off with doing chemical fixation in this project. And what happens in that case is that this tube, this membrane tube, starts to fall apart. So the actual structure we want to study in the electron microscope is gone. So it means we have to resort to cryofixation. And so cryofixation, we have two ways there as well. We can do plunge freezing, which most people use for structural biology, and you can do high pressure freezing, which freezes cells and tissue. So we can fix thicker samples in that case. And here we're looking at a cell, but we're looking at the cell where it's a bit thicker. That's where the nucleus is. So we decided we need to use high pressure freezing to fix this moment in time. So we can go from live imaging to an image in the electron microscope. And so what we did is we followed this workflow. This is, again, one of those schemes which you can follow. So we do live cell imaging, in this case, high pressure freezing, followed by freeze substitution, which is rather than doing the fixation and embedding at room temperature, we do this at -90 degrees. At -90 degrees, we add acetone, which is liquid at -90, that takes out the water from the sample. We add the osmium, so it fixes as soon as something is dissolved by the acetone, it is immediately fixed by the osmium. And then we slowly bring that to room temperature and infiltrate it with a standard EPOM resin. We harden that and we section that. And so that's what we do. Now, fluorescence, we cannot see directly in the electron microscope. There are some ways now to do that with integrated systems, but most of the time we have to use some other trick. So what we did is we coupled transferrin with Alexa 594, but we also added a 5 nanometer gold particle to this transferrin. We did the same for EGF, which was coupled to Alexa 488, the green dye, but in this case, a 10 nanometer gold particle. So now we can discriminate transferrin and EGF by color and by size. And what we did is we worked in this case with Leica Microsystems, and we developed a system together that's a high pressure freezer, but that allows the transfer of a sample from a light microscope into this high pressure freezer on a fast time scale, about 4 to 5 seconds. And so our sample sits in here in this carrier, and all we need to do is grab that loader and put it into the machine. And then the machine does everything automatically. It closes the system, shoots it along a rail, and then the sample is frozen. And that's done under high pressure, so we can have a thicker sample. As you can also see, this system is on wheels. And so it means that we can wheel it next to any of our light microscopes. So here we've wheeled it, in this case, next to one of our spinning disc confocals, and we start imaging. We have that loader on the table, and we can grab that and push it into the machine anytime we think is a good time point. So this is what we recorded. And this is not the best light microscopy, live light microscopy we've ever done. But that's just the technical challenges of having such a system. But what I want to draw you, so this is one cell that has taken up these two probes. And I want to draw your attention to this part here of the cell. You see that there's a green structure moving away, and that's where the movie stops or starts again, because that's the moment where we've taken that rapid loader and pushed it into the machine. So now we're going to trace back this event down in the electron microscope. So at the top, we have at -15 seconds, not much is happening, -10, -5, and only at 0, when we took that loader, that's where we see a bit that segregation of the green and red. That's what we fixed. We trace back that same cell in the electron microscope, and I'll come back to retracing in a bit. And we map the fluorescence onto our structural EM image. And we look at that in higher resolution. So remember, the transferrin was red and is coupled to 5 nanometer gold. I've put the red arrows towards those 5 nanometer gold particles. The green ones are EGF, which are a bit bigger, 10 nanometer gold. And now you can see that what looked to be almost starting to segregate is really at the structural level, really two or more distinct structures. So the transferrin is in this big one. EGF is mainly in these two ones. It looks like something is segregating here or merging, but that's what we don't know. And then we look at that in a better way, because this structure here looks to be connected, but we don't know for sure. Is this connected? That's where we have to combine this with electron tomography. So electron tomography takes a bit thicker sample, rotates that in the microscope, and takes images from different angles where we can reconstruct that volume. So that's what we've done here. And we've taken serial sections, and then we reconstruct this complete volume. So this is the structure again that contains the transferrin. This mainly contains EGF, and this contains EGF. So now we can see that all these structures are still interconnected. It's almost one fluorescent blob in the light microscope, but we can see that segregation happening. But in the electron microscope, we really can see much better the structural details. Also, what we found here is that when we completely trace the membrane, and it will come back in a minute, is here, there's a membrane structure pinching off from this transferrin endosome, and it's connecting to the plasma membrane. So what we think we found here is really a direct connection between the recycling endosome containing the transferrin and the plasma membrane recycling back that transferrin receptor. Of course, this is one example, and that's one of the big disadvantages of doing this technology. The throughput at the moment is still very, very low. So we need to be able to repeat that much more, where we, for instance, need automated analysis of such events that we can track them much easier. Those are all things we're trying to work on. But we had to use high pressure freezing or cryofixation in this case to answer that biological question. We know that, for instance, these membrane tubers that are here, they fall apart when we do chemical fixation. So the next project I would like to discuss is a bit different. And we took a completely different approach, much easier. Because that's where we looked, and this is a collaboration with Jeremy Carlton from King's College in London, and he's studying escort complexes. Escort complexes are involved in membrane fusion and fission events. And he was looking at dividing cells. So when cells divide, they break up their nuclear membrane, they divide, and then once the DNA has segregated, it reforms again, and then it rebuilds that nuclear membrane around it. And what he found is that on a cell cycle of about 24 hours or 22 hours, at only 90 seconds of that whole cell cycle, this escort-free complex is located somewhere around this reforming nucleus. So that's 0.1%. 1 in 1,000 cells will be in that state where escort-free is localized on that reforming nuclear membrane. Now, we can try to look at that in the electron microscope, but finding that 1 in 1,000 cells is really like finding the needle in the haystack. That will take you hours, days, months. So that's not feasible. So you have to come up with a different solution. And that's where we have quite a neat solution, and it's very easy. So for those, this is mainly a light microscopy meeting, so you will recognize the live cell imaging dishes. In this case, these are from Matec, and they have a special cover slip glued in the bottom because it has a finder pattern embossed in them. And here, for instance, it has fine numbers and letters. In this case, it's 5Q. This is where our cell of interest is. So now we can easily retrace our cell of interest by using those Qs from the letters and numbers. Here we've then embedded that sample and trimmed it down so that it really only contains this part with very few cells. And that allows us to really trace back those dividing cells. And this is done by Lorna Hodgson. She's very, very talented postdoc, and she does most of our correlative microscopy workflows. So what we have is, in this case, we have fluoronanogolds. So that has a fluorophore, but a 1 nanometer gold particle to it, because that penetrates the cell much, much easier. It means we have to then silver enhance those to find those small gold particles back, like here. And again, what we do is we do that tomography. So we acquire a tilt series, and all those black dots, that is where the escort complex is located. And so we see it on these vesicles, and we think those are incoming vesicles going to this reforming nuclear membrane. So the bottom part here, that's the nucleus. This is the cytoplasm. And you can see that it's located mainly at the nuclear membrane. If we then reconstruct that volume, and that's where we really go from top to bottom through that, then you'll see that you can nicely see the membrane, and then you see these dark blobs. That's where the escort complex is localized. And if you look carefully, you will notice that that's mainly localized at places where that outer membrane is still in contact with the inner membrane of the nucleus. And so that's what we think. And if we look at that in a bit more detail, go back. Then we can see this is again the nucleus. These are the gold particles. Those gold particles are almost always localized inside those small last holes in that nuclear membrane. So it's really bringing in, together with membrane, it's closing those last gaps in the nuclear membrane, sealing the nucleus again. These are not nuclear pores. We've checked for that. Nuclear pores are much bigger. They have a different structure. So this is really something different than a nuclear pore. They're reinserted in a completely different way. So those are two completely different approaches and using correlative light electron microscopy, each driven by the biological question. So the last part I want to talk about is something we've recently finished, and that's Cryo CLEM. So doing correlative microscopy and fluorescence at cryo conditions. And it turns out that fluorescence is very, very stable under cryo conditions. So again, this is a cryofluorescence microscope. What we do in this case, we grow our cells directly on an electron microscopy grid. We freeze that, plunge freeze that in this case, and then we analyze that first under the cryofluorescence microscope. And that is to trace back. Here we've induced microtubule-driven extensions of the cell by adding kinosore to these HAP1 cells. Now, we cannot search for those extensions, which you can see here on the left. We cannot search for those directly in the electron microscope. As soon as you put the sample in the electron microscope, in the cryo-electron microscope, you basically start to destroy your sample. So you cannot search very well in the microscope. So here we use cryofluorescence microscope. We make a map of that grid, and we can then see where those extensions are and trace those back in the cryo electron microscope. And this is where we were interested in the structural properties of microtubules. And so again, we do cryo electron tomography in this case. And this is again the reconstruction. And to our surprise, to our big surprise, we found there's material inside those microtubules. And it turns out that that's actin. And so that's something we found very recently, and we're now looking at whether these systems are real. This is, remember, this is induced. This is by adding kinosore to cells that drives these extensions here and maybe takes along actin. We don't know. So at the moment, we're looking for physiological systems, whether that's true. So again, here we just use the fluorescence to tell us where to look in the electron microscope. We don't use the live cell imaging capabilities of light microscopy in this case. So with that, I'm going to finish up. There are many, many ways to do CLEM, which I've only highlighted very few of those. But again, it's the biological question that drives what kind of technology you use. And so by now we've made a couple of books on these technologies where, again, it's described how you can apply those technologies. This one is coming out, and I'm very happy. Erin is one of the contributors to that book. This is coming out early next year. And one of the most important things is also to teach about the technology. It's those little tips and tricks that you only get from looking at the real thing. So we run an EMBO course on correlative light electron microscopy, usually in Bristol. It was planned for July this year. Of course, that didn't happen. And we're still thinking of ways to do that. Luckily, just before the lockdown, we did do a CLEM school at the Gulbenkian Institute in Portugal as part of COMILIS. And COMILIS is a EU cost network, really trying to bring together all the people doing not just CLEM, but correlated and multimodal microscopy. And with that, I would like to thank all the people in the group. So Lorna is really a postdoc that has done a lot of the correlative work. Judith is my electron microscopy specialist. I would be lost without her. Hugh and Jen have just left. So Jen did the Cryo CLEM, and that's now taken on by Chisato as a new PhD student working on that microtubule project. And with that, I would like to finish and thank you for your attention.
Thank you very much, Paul. That was fabulous. Really enjoyed it. I always love seeing the beautiful stuff that you put together. It's you make it look so easy, and it's not. So we have a few questions that have come in through both the chat and unsurprisingly, the first ones have kicked off actually from within the panel. So the very first question is from one of the selected talks from just after the coffee break, which is from Theodora. So her question is, do you get any question or any quenching of fluorescence dyes when you attach the fluorophores to the gold nanoparticles?
Of course, Theodora knows. I've just switched off my screen share. That is one of the things my previous PhD student, Hugh, was working on. So we know that if we bring a gold particle close to a fluorophore, it's going to quench. And actually, we measured that with an interference microscopy technique, and I've forgotten the name. What we actually found is that, and it's especially the green fluorophores that are quenched. So we took our EGF Alexa 488 with 10 nanometer gold, and we measured that. And to our embarrassment almost, we found that more than 90% of the fluorescence is quenched. So that is shocking. That's a commercial probe that we analyzed, and when there's gold, 90% of the cases there is no fluorescence present. What we
That's
do
huge.
see is a lot of fluorescence, but that is probably just the EGF Alexa 488 that has lost its gold.
So do you think that's size dependent? Do you think with smaller golds?
Yeah. There are already studies by an American group that have looked at that. So it's size dependent, and it is fluorophore dependent. So the green ones are, but that's the worst combination we could have chosen, is a green fluorophore with a big gold particle. That's published some time ago, about 3 years ago. We published on that because we were using the probes. So we thought it would be better to publish ourselves that those probes are not the best to use. So we think that's why we think, for instance, the fluoronanogold is a very good probe, because that's only got very small gold, and we use the red variant of that one.
So small and tidy and very good. Okay, so the second question, also from within the panel, and my co-conspirator, my co-chair, Javier, he asked, for the EGFR project, once you record the fluorescent images, how do you, this is such a simple question, but not, how do you relocate the same cell in the TEM?
Okay, so we have different retracing systems. With the high pressure freezing system, we have to grow our cells on sapphire discs because glass doesn't freeze very well. So what we do there is we carbon print a finder pattern first on the sapphire disc and then grow our cells on there. When we do the freeze substitution, the carbon pattern lifts off with the cells. So again, once we have the block, we can trim it down exactly to the size of the cells we need. And so there's, for the cryofluorescence, for instance, we use already finder grids. So again, we record, those are things, it was on the very last slide where you can see the numbers again, which we use for retracing. But maybe also further on, of course, you need to map the fluorescence image on the EM image. You can do that in Photoshop. That's the easy way. Again, it's nicely described in one of the chapters in the books. A more sophisticated way is by using software called CLEM, which is developed by Perrine-Paul Gleteux in France, and that really sort of say looks for reference points which are matched, which then can take into account some warping, some shrinkage, and things like that.
There's a chat that just says CLEM rules, and I couldn't agree more. It's a great program. Yeah. Another thing I think you can also do if you're dealing with 3D is you can try and venture into the world of a Mira as well, which can be really, really helpful, although its own fight in and of itself. So there are different tools out there. Yeah. In the Meet the Speaker, I thought if there aren't a lot of questions, what we could do is have a discussion about how you would set up one of these protocols, a CLEM protocol, and how you would sort of approach how to answer these questions, how to do the stitching, some tips, just chatting, right, Paul? We always can talk for hours.
We do.
There's one last question that I think we have time for from Sonia. Sonia, who gave a great CLEM talk on Tuesday. I don't even know the days of the week anymore. They all blend. It was Tuesday. She gave a great talk, but her question for you is, for the actin inside the microtubule story, which is super cool, have you looked at cilia? That's her pet structure, just so you know. Yeah. She's curious because she wants to know when looking at multiciliated cells, whether there are multiple ciliae within the same plane, if you also expect the actin within those structures.
The answer is no, we haven't looked yet. So we've just started with that. At the moment, we're looking at megakaryocytes. So they these cells make the platelets, and they make long extensions. That's where that's what Chisato, we've just acquired data yesterday on that. So we're analyzing that. Another system we're looking at is neurons, because of course these have long extensions. With the cilia, it's we have to get them in the right plane. With cilia usually sticking out in different ways, we couldn't think of a good way of tackling that at the moment. We are going in, so to say, into volume EM, looking at those things. But again, if you're going to do volume EM, your resolution is going to be too low to really map actin in there. That's still, some people still debate whether what we are seeing is actually actin, because they haven't seen it. But I think we've done a proper analysis on that. People have seen, so to say, material inside microtubules for a long time, but that's on chemically fixed material. So you can't, so to say, do the real mapping of those.
Exactly. I think my reading of Sonia's question, now that I looked at it, is also she was hinting that with a multi-ciliated cell, you'd have more chances of getting the right plane, and she's probably right. The more chances we have in these things, it always helps.
Yeah.
So for the interest of time, Paul, let's move on. Your talk was fabulous. Thank you so much for joining us here, and I look forward to more chatting when we get into the Meet the Speaker